right? So if you got a nonsense mutation in a ribosomal RNA, so what? As long as the ribosomal RNA is still able to fold and do its job, so what? But we were talking about RNAs that code for protein. If you're in the coding region, then a premature stop codon would likely lead to a non-functional protein. Okay. Now, missense mutation, we said there are a range of possibilities, right? Missense mutation could lead to no change. It could lead to a non-functional protein. It could lead to less function. It could lead to better function. It could lead to new function. And it all depends on what amino acid has changed, where in the protein has changed, and what the effect that has on the functioning of the protein. Okay? Questions about any of that? Okay. We did introduce one additional type of concept, which is a conditional lethal mutant. And that is a mutant that has two types of conditions that affect it. One is permissive conditions. And so under those circumstances, both wild type and mutant can grow and reproduce versus restrictive conditions where only wild type can reproduce. The mutant might be able to grow, but it can't reproduce. And what example did we talk about for a conditional lethal mutant? Temperature sensitive, that's right. So temperature sensitive mutant in which the wild type can grow at 37 degrees. The wild type can grow, the mutant can grow. Sorry, it's getting late. Wild type can grow, mutant can grow. But at 42 degrees, wild type can grow, but the mutant cannot. And what's a likely type of mutation that led to that type of phenotype? Missense. Why do you say missense?
It could change the amino acid that could cause it to fold differently and behave differently at the higher temperature.
Right. It could make the protein less stable at the higher temperature. At the lower temperature, you wouldn't notice. So that's why it's considered conditionally lethal. Does that make sense to everybody? Okay, now, in this area, we talked about these different kinds of mutations and how we could revert them. Okay, so that's the next section, was reversion of point mutations. And again, when we talk about reversion, what we mean is to the original phenotype. That's what we're talking about in terms of reversion. Not necessarily to the original genotype. So we looked at examples where you could get back function even though you haven't actually gone back to the original set of nucleotides and organization of nucleotides. Okay? So for that, want me to walk through those?
Can you just give us like a brief overview?
Sure, we'll try and make it real brief. Because after that, the only other thing we talked about are the specific examples of larger mutations. Okay, so let's say we have lac z plus. And lac z plus, its messenger RNA looks like this. So you've got AUG, GUC, CCC, ACA, UCA, UGC. Okay? And if you get a mutation here, so that this C is now a G, then what's that codon? Stop codon. So that's going to become likely lac Z minus, and that's because this now says stop prematurely. Okay? Now, if you wanted to revert that, how could you revert that particular mutant?
Missense mutation?
Well, what's the most direct way?
Inserting the original nucleotide.
Okay, and that would be called, can I true revert? That would be a true revert, exactly. So if this goes back to the original, that's the true reversion. Okay. But what that implies is there's other ways to do the reversion as well. And so the examples that we talked about were to make some other change in that G that doesn't give us a stop codon. So you can change that G to what?
U.
A U, that's right. So what if we instead change this to UA? Now this is not stop. So it codes for some amino acid. Is that going to be the same amino acid as UCA? So this will be a different amino acid. And so that would be what kind of a mutant would become? That would be a missense. But we already know that missense mutations can be silent. And if it were silent, then this would be also a possible lac Z plus revert.
Quick question, under the stop, does that say UC or UGA?
UCA, this one is going back to the original.
Okay.
So that's the true revert. Okay. But this UUA is a revertant, a hypothetical revertant. It may or may not actually work. You wouldn't see it, right? If it doesn't work, you won't notice. If it does work, if that missense is silent in terms of the function of lac Z, then you would see that as a revertant. Okay? And then we went through and said, yeah, basically you could change the U, you could change the A. The one thing you can't do to make this work is to change this G to an A, because then you have UAA, which is also a stop. Okay? But the idea is that you could change any of the nucleotides in the codon to get rid of this from being a stop codon and turn it into some other codon for an amino acid. All of those are potentially missense mutations if you do it that way, but any or all of them might do the trick. It just depends on the protein, where it's happening, what the protein needs in order to work. What you know for sure is if there's a premature stop codon, that protein is not going to work. Okay, does that make sense? Cool. Okay. We talked about if there were a missense mutation. So let's say that we have a different mutant. This mutant, we change the U to a C. So this is going to be a missense mutation as well. And what we can hypothesize is that that is also lac Z minus.
How do you know that's not a silent mutation?
I'm just hypothesizing that it's not. So for purposes of this argument, let's say that that is not silent. It could be silent, but let's say that it isn't silent. So if it's not silent, then we have a laxy minus phenotype. And then how do we revert it? Well, all these same ways we did here, but there's a different way you can revert a missense mutation that doesn't change this mutation, but relies on a change somewhere else in the protein. Question?
I was gonna say, like, changing the tRNA.
That's a different thing.
Okay.
We'll come to that. And there's a name for what you just suggested, which is what?
Suppressor.
A suppressor. So that'd be an example of a suppressor, because you get a mutation in a separate gene that controls the original phenotype of your first mutation. Okay? And not in this class, but in some classes they'll refer to this example I'm giving you now also as a suppressor, but rather than confuse the issue, let's just say that this is just changing a second codon somewhere else in the protein allows for this missense mutation to work if you have a second missense mutation somewhere else in the gene. So the idea is that you have a wrong amino acid here, a wrong amino acid here, but both of those wrong amino acids allows the protein to fold to give back function. Okay? Does that make sense? Okay, now, the example that was just given though, what if you have this mutation and from that mutant you select a LAC-Z plus revertant? That retains the nonsense mutation. So if it retains the nonsense mutation, that should be a problem, right? But the suggestion for how to fix that is you get a mutation in a separate gene, making this a suppressor mutation, and that gene that's recommended was the gene for a tRNA. So if you change a gene for a tRNA so that the tRNA that's made recognizes this stop codon, then you're able to go past that stop codon, and that mutant tRNA will put in its amino acid. So you're still faced with the problem of, well, can I put in the wrong amino acid here and have the protein work? So it's still a situation where you're not really making a true reversion, but if you put in an amino acid here instead of stopping, at least you can make the rest of the protein, and maybe that'll work. Does that make sense? Okay. Any questions about any of this point mutation stuff?
In regards to the exam, we're supposed to explain all of this in words?
Maybe. Yeah, I mean, you'd have to, kind of draw this out and say what you got going on there. Or maybe you'd be given an example like this and have to explain how or why to fix it, if that makes sense.
Okay.
Yes. What was the deal with the GCC at the bottom?
GCC. So in this situation, the mutation is not a stop codon, it's a missense mutation. And so that could be LAC-Z minus, hypothetically.
Okay.
All right. Now, if it is LAC-Z minus, hypothetically, you could revert it by all the means we talked about over here, except for this one, of course, except for the suppressor. But what if you retain, so now we're going to get a revertant. What if you retain the missense, and now you have a missense somewhere down here? So now you have, this is one wrong amino acid. This is a second wrong amino acid. So now you've got a protein with two wrong amino acids, which might allow it to fold in such a way that it regains function.
Okay.
Yeah. Well, I mean, if you think about how proteins evolve, the hemoglobin that we have is not identical to the hemoglobin that, say, a chimpanzee has, but they still work. So there are amino acid changes that have occurred over evolutionary time, and yet they still let the protein do what it needs to do. Okay? Yes?
So GCC is not a silent mutation?
In this hypothetical example, no, because it made the protein not work anymore. I mean, you could come up with a different hypothetical example where that might be just fine. But I'm suggesting that by making this change, that makes it lax and minus.
But it could also undergo like another missense mutation, producing like,
Yeah, so
reverting back to original condition.
yeah, if you get the second mutation, in addition to that one, you get function. But I would also hypothesize that if you get this mutation by itself, without this original mutation, that would also be non-functional.
Can the GCC undergo another mutation?
It could, but then that would be just doing what we did over here.
Okay.
Yeah. So I was just trying to show you alternatives.
Alternatively, okay.
Yeah. So you could, in this case, you could try to revert this in all the same ways that we did over here, except for the mutant tRNA. But yeah, so that's what you can do. But then you also have this additional possibility for a missense mutation that you don't really have for a nonsense mutation. Okay? Okay, the last thing we talked about, and you guys can tell me how much of this you want to go into, is mutations involving more than one base pair. So we talked about examples of deletions, rearrangements, and increases in amount of DNA. So for deletions, we talked about Cree-Duchat. Everybody remember that or need to go over it at all? Okay, so that's a deletion of part of one of the chromosomes. We talked about Fragile X. Again, deletion that's due to, ironically, it's due to an increase in a certain repeated sequence, but that makes the X chromosome fragile in that particular location. The bigger that repeat gets, and so part of it can break off, and that's the problem with that one. And then finally, this is the biggest deletion of all, monosomy. So this is complete loss of an entire chromosome. And we said that that is usually, in mammals, lethal, right? Except, that's right, except Turner syndrome. Okay, which is one X chromosome and no other corresponding sex chromosome. So no other X and no Y. Okay. All right, so that's those. Do we need to talk about them at all? Great, okay. Then we got rearrangements. So we had inversion. So that's where a region of the chromosome is inverted, which may or may not affect the individual, right? Because it depends on where the breaks go. But is likely to affect offspring because it's likely to affect meiosis. After recombination. Okay. So that's one example. We also talked about transposition.
Can you talk about anything about like a variant gamete formation?
That's what we're talking about.
Okay.
Yeah. Transposition is where a piece of DNA jumps to another piece of DNA. And this is the sort of jumping genes. And the problem is it's likely to interrupt a gene in a new location. Okay. Now that's different from translocation, where a chunk of a chromosome is exchanged with a chunk of chromosome that it's not homologous. So, the example, yes, question?
So in the transposition one gene loses a portion of its DNA because it goes to another one, while in the translocation, it's like both gain and both lose.
I would say for transposition, you can lose the,
Oh yeah, you can either keep it there or copy and put it into something
right,
else.
so there's kind of a cut and paste versus a copy and paste approach to transposition. If you copy and paste, you're going to keep that place where it was originally, but the new location that it goes might, which is essentially random, might then be a gene that gets interrupted. Whereas with translocation, you're actually physically taking like maybe one arm of a chromosome and breaking it off and reattaching it to another arm of a different chromosome that it's not homologous to. In both cases, the movement of DNA is not between homologous chromosomes necessarily. Whereas in translocation, it's definitely not a homologous chromosome. So transposition could be from one homolog to another, but translocation is always from one chromosome to a non-homologous chromosome. Okay? All right, so we talked about the example of translocation being familial Down syndrome. Do we need to go through that, or would you like me to go through that? Sure, why not? Okay. All right, so the example we talked about is, so this is familial Down syndrome. And what you have is a cell in which you've got one copy of chromosome 21. You've got one copy of chromosome 14. And you have a translocation chromosome, which is chromosome 14 with most of chromosome 21 attached to it. So this is the translocation chromosome, and an individual that has this type of situation is referred to as a translocation carrier. No phenotype difference. So this is silent in the sense that you wouldn't know it if this were, if all of us or any of us in this room had that, you wouldn't know it by looking at us. There would be no outward phenotype. Okay? But when this individual produces gametes, we know that each gamete should have one copy of 21 and one copy of 14. But this individual can produce different kinds of gametes than that. So this individual can produce a gamete with 21 and 14, which would be what kind of a gamete? Wild type. But it could also produce a gamete that has 21 and the translocation chromosome. And if that were true, and that individual were to mate with a wild-type individual, what would they produce? Down syndrome, because they'd have 21, 21, and 21 from the wild-type gamete. So they'd have 3 copies of the 21 information. So that would give you Down syndrome. Now, because of the way that works, either 21 gets to go with 14 or it gets to go with the translocation chromosome, and there's only one of them to go. So if it doesn't choose to go with 14, that gives you a gamete with just 14 and not 21, which is
Fatal.
That's fatal. It's fatal to a point where that particular offspring will die in utero. Okay? Because that's monosomy for 21. So if you only have 14 and you don't have 21, that's monosomy for 21. And then the other possibility we said was you could get just the translocation chromosome, and then when that gets fertilized with wild type, you have
Translocation carrier.
you have what you started with. Okay. So the reason we call that familial Down syndrome is this individual has a high probability of producing either a Down syndrome offspring or a carrier. The Down syndrome offspring is not as likely to reproduce, but the carrier is quite as likely to reproduce as wild type is. And so that means that you're going to have a family that has, from generation to generation, a large number of Down syndrome individuals. Okay.
So this is a silent mutation?
Sorry?
Is this the silent?
I wouldn't, yes, but I wouldn't,
Oh, okay.
yeah, I think it might confuse you more if you call it a silent mutation. I would say that there's no outward phenotype of this individual.
Okay.
All right, and then, so that's rearrangements, and then finally, the last one is duplications. And I think we just talked about 2 kinds. So one was aneuploidy, and the example was trisomy 21 due to non-disjunction in either meiosis 1 or meiosis 2. And the result then is that you get gametes that have 2 copies of chromosome 21, and when they're fertilized by wild type, then you have 3 copies, and that's how you have trisomy 21. Now, when does that show up most often? Sorry,
What? Can you just repeat that?
I'm sorry, I'm not hearing.
Could you repeat that?
Yeah, what's the question? Yes, the question is, when is that type of trisomy, and by the way, this is 95% of Down syndrome cases.
Also, older women.
Sorry?
Older women.
Yeah, older women, when they give birth, their eggs have been around for as long as they've been around. And so effectively, any mutations that have accumulated over the person's life could affect disjunction during meiosis. That's one of the things that could happen. And so that tends to be more likely in older women that are giving birth. Okay. So that's aneuploidy. I don't know if anybody wants to talk about anything else about that.
Do we need to know how you can get the monosomy, like in class when you drew out the chart, and how you get monosomy and how you get normal phenotype?
You mean in familial? Oh, you mean
For
in
traditional.
well, I think what you can try to explain in words is that you're going to get non-disjunction in either meiosis 1 or meiosis 2. So if it's in meiosis 1, then you're going to have 2 cells that are going to be without 21, and so you get monosomy. Versus if you do it in meiosis 2, only one of those cells is likely to be without a copy of 21. And so you have to kind of think of a way to say that in words, even though you can't draw the picture. I'm sorry, but it's a defective program, in my opinion. It's not the lockdown program that's defective, it's the test program that lockdown program uses that doesn't allow you to draw, which I think is dumb.
Can it not like record us while we're writing stuff down?
Sorry?
Can it not like record us while we're like writing?
Record you,
Yeah, because essentially it's like a webcam thing, right?
like record you talking?
No, just record us like doing tests.
Oh, it is gonna record you.
Right,
Yeah.
right. Can it not do that when we're like writing in like the blue book, for instance?
Oh, well, you're not gonna have a blue book, you're gonna be,
Well, yeah, but I'm saying like, is it not able to do that? Can you not like use a blue book while it records it? I'm curious.
Then you could take your blue book later.
Yeah.
yeah, or you could, I suppose you could draw a picture and show it on the webcam.
That's what I was thinking.
Oh,
Yeah.
can we do that?
But then you have to watch the video. It would flag
Right.
every single one of us.
That's
Yeah.
Every time.
Oh,
Yeah, that's the problem, is it would flag you guys, yeah. I mean, which, you know, if you want me to look at all of your videos, it'll just delay how long it takes for me to grade your exams, but that's okay. Yeah, it is a problem. And what I hope is the next 2 exams after this, it's not an issue. But we have to deal with it. Yeah, exactly, we have to deal with it. And we're doing the best that we can, all of us are, I'm sure. So, okay, so that is one example of a duplication. The other one is just a gene duplication. And I don't remember how much we talked about this in class, but I particularly like the gene duplication example because it helps us maybe understand how a suppressor mutation can occur. So if you have a gene, so let's say you have a gene for a tRNA. tRNA, your favorite gene, or your favorite tRNA, whatever it is, for some amino acid. Okay, here's the gene. And then if a mutation were to happen in that gene by itself, that can be problematic, right? Because you need that tRNA for the cell to be able to do its job. Excuse me a second. But what if you had a duplication of that gene? So now you have a gene for the same tRNA. Okay? So this is a gene duplication. Now, what might be a consequence of having a gene duplication like that?
increased expression.
Yeah, increased expression. So now you've got twice as much tRNA than you normally would have. Now, there may be a negative consequence, there may be a positive consequence, there may not be any consequence. It just depends on the particular cell. Having twice as much of that tRNA, you may not notice. Or it may make the cell more efficient for using that particular tRNA for that particular codon. Or it might be bad. You know, maybe there's it might now prefer to use that tRNA than another tRNA for the same amino acid. You don't know. But the point is, you've got 2 copies and both of them work. Now, in reality, the cell only needs one of those copies, right? Because it needs a functional copy so that whenever you need this particular amino acid put in, you've got the tRNA for that during translation. So that means that if you only need one of these copies, the other gene, the other copy, the new copy, can start collecting mutations. Okay? So this copy can collect mutations. Now, that mutation might make this tRNA completely non-functional. But that's okay, because you have the wild-type copy over here. Okay? On the other hand, if that mutation changed the anticodon so that now it recognized the stop codon, now you've got your original gene for a tRNA, and you have a second copy that provides a new function that you didn't have before. And that's how you could get a suppressor mutation that deals with that nonsense mutation we talked about before that didn't change, but now you've got a tRNA that allows you to fix it. Okay? Now that's, again, this is hypothetical. Not every duplication like this and every change in a tRNA is going to do that. But you can imagine over evolutionary time, there may be a selection for having that occur so that if you were originally lac Z minus through that nonsense mutation, and that happens, now you can become lac Z plus again. Okay? So there's a selective pressure that allows those instances where that occurred possibly to have a good effect or a positive outcome for the individual. And that's it. And those of you who recorded this, I appreciate your recording it. I also appreciate if you can contact me and your peers in terms of uploading this to your respective sections. And also, in your case, you have the visual recording as well. So if you could send me a link for that, I will give that to the other sections that you're not in.